figured we do something like
That's the
that.
first thing to try.
What's that? Switch. Okay. Looks like this one. Yeah. I don't know. What you said? This one in the box here. I think it might be the one, that brand new box. I think that's what it is. Oh, it's not a wireless mouse. That would explain it. Is it a wireless mouse? No, it's like it. good. Oh, you know, do you have any, is there any packaging tape, like clear packaging
I think
tape?
I have some. Let me go check. And I'll be
Great,
right
we
back.
won't need it too much later, just thinking
No
ahead.
worries. Yeah, let me go grab it and I'll leave it here.
Thank you. Great, thank you. Oh, perfect.
You're welcome.
Thank you very much. You did? Well, let's see how far we can go that way. Get it out of the way. Yeah, the rest of this crap in the box, out in the hall first.
Interesting stuff.
Sorry?
Interesting stuff.
Yeah? Yeah?
Have you used any of these tools before, software or hardware?
No.
No?
Back there, yeah.
Yeah, right, yeah. So I took a look at Spring early on.
Yeah.
Guild.
Yeah?
I thought it was actually pretty easy to use.
Okay.
That shell, you know, I mean, if you do MATLAB, I mean,
Yeah,
it's
yeah.
pretty straightforward from that. Yeah, I found is that, you know, you have to find the right questions to ask. Right?
Yeah.
How to get that information out, but
Yeah.
a lot of it's there, which is good.
Yeah, I guess as long as he's just a user, more or less.
Yeah,
Then
yeah, yeah.
But
Yeah.
no, looks like
I
interesting.
find-
Yeah, we start to see a lot of papers.
Yeah, right. Yeah, it's just sort of very early days, I think, and, you know, a lot's gonna happen next many years, so.
Yeah,
There's definitely a different way of looking at cells and tissues, right?
yeah, yeah.
And biology.
Yeah. Well, I'm immunologist, so I'm kind of late for those, but still there's genes, so, you know, it's expressed.
Right, exactly, yeah. Yeah. So it seems there's a lot of overlap with stem cell research.
Yeah,
so
yeah.
it's very,
That,
Some
and I
common
guess
areas.
again, some of cellular more like variable,
right.
context dependent.
Mm-hmm.
Expression.
I'm also beginning to see some new methods coming out for, there was a question yesterday about spatial location.
Yeah.
And so I think it's pretty key.
Well, yes, but then everything gets more. I think
Time
it's just-
and state,
Yeah.
and also in different states if you can.
Isn't how it's called?
So we start looking at some
Uh-huh.
these new methods and see if we can incorporate it to the kind of things we're offering. So, so you
Like
can get-
directly.
Yeah, so, you know, there's various force of probes
Yeah.
developed, and I guess I'm not quite certain, this is kind of new, sure you understand how it works and how stable it is, and, you know, reproducible, reliable, but, you know, people use transcriptional signals you saw with Alon's work to locate to a position. You know, there's work out of the Broad Institute on that, to try and put this piece of information together, that's more inferred,
Yeah.
than anything else. I think that's like the next big push with the other sort of other measurements on singles.
Yeah. That, yeah, I guess we have to funding for getting more instruments, I guess.
And more
So
exotic
it's the
things
same
to do.
problem, yeah. It's like, it's always the same in a private university, we have all those donators and all things. I mean, it's
Yeah.
cool, but I mean, to use it, you need money or something.
Yeah, yeah. I think also what I see is the know-how, right? And, you know, how do
Yeah, well,
put it
you
all
depend
together?
more and more on people, obviously. I mean, many of those technologies. If you have in your lab, except if you're a user,
Yeah.
cannot spend time on just. I think it's the same with imaging, and it's becoming more and more, so high tech that really dominate.
Right, right. Yeah.
You know, you'd use like 5% of it. Or something.
Yeah. But it's really, you know, I find, you know, I mean, I'm sure you've seen your field, it's getting more multidisciplinary.
Yeah,
I mean, discussion yesterday was primarily around bioinformatics. You know, how do you take these massive data sets
Yeah.
and figure out, you know, get some answers
Yeah, I know
to
that's the other
questions.
side, because again, you know, mentioned that. I've just downloaded that, because I'm not using it, so, and, I mean, all those things, I mean, and deep sequencing, all those things, you know, if you get the process that, okay, and the first part, you know, and all those things,
Oh, yeah,
you depend on,
yeah, yeah. I say that, so my background is physics and engineering.
yeah.
And I was at MIT for many years before
Yeah.
going into my entrepreneurial thing.
Yeah.
And, you know, I found, you know, when you converse with tools and such, you know, it's not as big a deal. I noticed going into now in building instrumentation and such, and, you know, offering it to folks that there's a gap, there's a skills gap, there's a knowledge gap.
Yeah.
And, you know, it's becoming, I wouldn't say it's becoming bigger, but certainly there's more things you have to do. And you're right about the specialization and the team effort, because they're,
I mean, you know, and
J'en ai
then
pris
it's
pour
not
une
easy to
fois,
get, you
d'habitude,
know,
dans
like,
ce
for
sens,
example,
j'en
we
ai
have Rochester
Mais,
Institute of
en
Technology,
plus, là, j'en
they
ai
put people
rien
doing
à faire à 2
computer,
heures.
whatever. But, you know,
Gamberger. Fermé, mais-
when they decide whether they want to go to research or to another job, well paid, you know, it's always make a big deal. So it's not easy to get connection.
that's what I came up. It's like somebody doesn't get hit by a bus. That's not a disaster.
Because I mean, you know, it's almost, I guess if it's good if you can have a team technology, you know, and start to know a bit about the project. I mean, otherwise then those dialogue problems. You
Yeah, I've run into that, you know, building teams, you know, for product development, where you have to have that breadth, you know, the technology and then the application side, make sure everything meshes together. So, it's always.
I agree. It's, I mean, I'm doing a lot of things more like collaboration with people doing ecology. So they have a lot of, you know, I guess more background in things like that. So that means that usually that's the channel for music.
Yeah.
Difficulties that like the processing data by others. But yeah, sooner or later you need to kind of start to really to understand where analyze the data.
Exactly, yeah, coming from this background, so everything's reduction, reducing molecules interact with each other, so it's all, you know, it's somewhat deterministic at some level, but here it's just a high complexity.
Yeah, but I think in order for sooner or later you have to go back, test that with an experiment.
Yeah.
Yeah, there's an idea. You go back, put your hand in it.
Yeah, how do you validate the data too? That's the other thing I
Yeah.
worry about because, all right, I remember the early days of microarrays. It's a hot area, right? And so many labs just jumping right in, generating crap, you know, because they have quality control metrics, you know, there's a whole bunch of things that, because they're, yourself, so when Afrometrics came in, it kind of started to standardize. That's when I thought you saw some advancements, because then people could compare between labs, sets, and start to.
Although still, I guess, there's still some problem.
Oh yeah, nothing's perfect, but you know. But at least,
I still remember the comparison between lab we mentioned. It was like
interlab comparisons.
there was not that much of a lab.
Yeah, well, that was the Mac QC project, microarrays, QC, and where I was in on that. One of my early companies we built a lot of PCR array. So array, but using PCR. Part of that project, and you know, coming up with various standards and have interlab comparison. It's a huge challenge. Because, you know, you don't get brownie points for doing things like that. But if you don't do that, then science can't progress because, you know, people start flummoxing like this is it real or is it an artifact? Interesting. So if it was easy, then, you know, why would you be doing it?
No.
So you're at Rochester?
Yeah.
And you're on the lake, is that where it's?
Sorry?
Institute on the lake itself?
No, it's like just maybe 10 minutes from the lake.
So what's it?
It's close to the airport.
And it's close to the University of Rochester?
And there's a medical center.
Oh, right.
Okay, let's try to get started.
Maybe that's the key. Okay, so that's the
If there is 1 hour this whole week that you do not want to miss, it's this hour. Get as caffeinated as possible and stay alert through this whole hour. So we're also recording this, but getting through video of this hour, it might not be the most efficient way if you want to get back. So I was gonna ask maybe 1 or 2 volunteers who are particularly good with taking notes. Take notes thinking that you might share this later. Caleb also is creating, README documentation and all of that, but maybe if we have several versions from a user perspective of, good practices and hints as you go, take notes and share it with each other, with everyone, okay? Thanks.
So, is everyone able to reach this website? Yep.
Who has not reached this website? Raise your hand. All right, everyone is okay then.
Can we get a microphone?
I'm
No,
Caleb.
no, I'm
Caleb.
Put it up higher, even though you have a good strong voice.
Oh, yeah, but it takes time.
On camera, there is a message that says
Oh, okay, out of batteries. Hold on.
They want to know.
Should everyone just try clicking on one of these stages? Avoid this all stages. That's just don't go there. Too many cells. But maybe just click here.
Which one was that?
That was stage 12. And just raise your hand if you, for example, have your screen is all black. That's happened to a couple people. That's happened to me. So if
No,
your
it turned colors.
Okay, if anyone sees a black screen, then switch to Safari. You have it. Okay.
Some people may see.
Raise your hand if you don't see a screen that looks like this. Okay, great.
I'll just leave this here and let it float, okay? Just pass it to one another.
Okay, so should I just start? Yeah? Leon?
Yes.
Okay, so welcome everyone to the official spring tutorial. I want to make a couple comments before starting. One is that the interface you're going to use today was co-developed between me and another person in the client lab, Sam Wallach. So just, you know, when the textbook editors come to ask you about this Jamboree, make sure you note the contribution of Sam Wallach. Second is that this is a work in progress, so it's going to break. I apologize ahead of time. Third, we just got a grant to improve Spring, but we don't exactly know what to change about it. We just we just kind of applied opportunistically. So if you have any feedback, and this is a whole grant we're talking about, so it could be very whimsical, ambitious feedback, then please email me or come to me in person, because it will be instrumental in guiding what we do with the grant. Finally, I posted my phone number in the Slack channel, and you should just consider me your IT specialist for the week. So if anything breaks, or if there's something you want to do and you don't know how to do it, then just text me. So the way that we're going to do this is I'm going to show you some features of the browser, and I want everyone to, as much as possible, follow along on your own computer. And we're going to take a break in the middle for some independent exploration, and then we'll come back together. So first, before we actually delve into the browser, I just wanted to say a couple words about what is, so actually let's all, so if everyone's here, this is a main page. As Leon said yesterday, this is the data up here is the complete set of cells in each stage, and also the tree view, and we won't be changing it. But down here are all the spring plots that you yourselves will generate, and we'll get to how to make those later. So could everyone just try navigating to one of these spring plots? What? Oh, yeah, if you want, you can turn off the edges. I'm going to keep them on for the sake of explanation. So the points I'm about to say have been explained a couple times, but just for emphasis, I'll repeat them. So, which is basically, what are we even looking at right now? So these, I clicked on stage 12. So these are all represent single cell. Each dot in this plot is a single cell transcriptome from stage 12. The way that we got this plot is through a couple bioinformatics steps from the raw data. So from the raw data that we get out of the sequencer, you have to align it, obviously. From there, you get a count of how many mRNAs you see in each cell. Then there's some steps of normalization. There's a step of and at that point, you can think of each cell as being a point in a 20,000 dimensional space. So from there, we do a couple steps of kind of denoising and other things to get to the point where we can construct this graph. And the graph is basically a statement about which cells are similar to each other and which are different in their transcriptomes. And the graph is our best summary of the kind of high-dimensional connectivity or landscape of cells. From the graph, we then need to take the graph and put it in 2 dimensions so you can look at it. And the main thing I want to emphasize here is that the precise geometry of these structures is not important. It's not, or I mean, it's very non-unique. So the fact that this spike is going off to the left is absolutely meaningless. It could have gone to the right. You know, a lot of the precise distances between things are completely meaningless. The coordinates of this 2-dimensional grid, of this 2-dimensional space, are in every way utterly meaningless. Please don't interpret them too carefully. Basically, the only part of this diagram that really meaningfully corresponds to the data is the structure of the graph itself. That is the summary of the high-dimensional structure of the data. And we've just done our best job to put this graph in 2 dimensions so you can see what it looks like, but the precise 2-dimensional layout is not unique. Mm-hmm.
So because you said the position is meaningless, basically, but the distance is not, right? If you look at the blue one there, it's very far away from the green one there. So these cell types, would you say that they are less related? So the blue one is less or more distantly related to the green one than maybe to, I don't know, red one or something like this. But it would be also distantly related to the yellow one, although they are next to each other.
So you can do a little bit of interpretation, but I would and yeah. So I totally agree with what you're saying, and so certainly a lot of information can be gleaned from the 2-dimensional layout. But I get a lot of questions like, what do the X and Y axes mean in this? And the answer is nothing.
And the cells in the center are more transcriptionally related to each other. Right?
Yeah.
Maybe this would help. Just a quick demonstration, right? If I have a 3-dimensional object, which is this cube I'm talking to, and I want to represent distances between vertices on the plane, this is about the best job I can do to faithfully represent all of the pairwise distances. And it's never going to reflect the truth, right? There is a necessary distortion. But here, we're going from very high-dimensional space into 2D. Okay, so it's important that this makes sense to everyone.
I'm just curious, what do you call this? So when we can describe T-SNEE's plot and diffusion plot, and so just so we can talk about these different ways of visualizing the data, what do you call this?
If you want to communicate with someone who's never heard of Spring, you would call it a force-directed layout. And in the lab, we call it a spring plot. Okay, are there any other questions about the meaning of the layout in terms of how it relates to the biology? Okay, so now let's get to actually how you use this tool. So the main thing that you're going to do here is look up genes. So let's take SOX2, for example. Or you can look up your own favorite gene. So you want to type SOX2 or whatever gene you want up here. So now the color is showing you the level of expression.
How do we do the zooming? Sorry, that's how do we do the zooming in?
Yeah, the same way you would scroll up and down a web page. And if that's not working for you, just make a note and I'll come look at it in a little bit.
Yeah.
Also, in general, if you find any of the features here not working, just try switching browsers and just try a couple browsers.
Zoom in out works for everyone. If it doesn't, raise your hand.
Zoom out means that bar with 30 number?
No, zoom in out just means this.
Okay, who is unable to do this? Raise your hand and keep it up until somebody stops by and helps you. James, can you help me?
All right, so moving on. So what does this bar mean up here? So basically every cell is so in this case for SOX2, in our original data matrix, every cell has some number for SOX2. The number of unique mRNA, the number of unique cDNAs that map to SOX2. But the color here, the intensity of color relative to that number can be changed by moving the slider. So if I move the slider to 10, then anything above 10 and above is all being colored the same. It's just being colored bright green. So this is, I think that's pretty clear.
TPM?
Yeah, what are the units, or what's the...
The units here are, so they're unique molecular identifiers. So it's UMIs per cell, and but these data have been normalized to correct for total count variation between cells. So you should think of it as just heuristically representing unique molecular counts per cell, but not in a precise way.
I'm looking at my own favourite gene, but my scale only goes up to 10. Does that mean it's not really expressed very much?
Yeah.
How do we know what is not expressed very much and actually doesn't count? Because it's so bad.
So the range of values shown here is always the minimum and maximum level of expression of that gene in the data.
Well, I guess my question is, if you can get a colour when it's on 2 or something, does that mean that it's really not really expressed, it's unreliable? Or something?
I think that the best way to decide if it's reliable is based on the appearance of the expression. So if it's very salt and pepper, if it looks kind of random and there's just a couple cells that are really bright and the others are dim, then that means that you can't trust it very much. But I don't think the absolute number is as indicative.
So why do some have a yellow circle around and others don't? In my view, at least.
So you may have clicked this. If you click this, that'll now happen. If you click it, and you can deselect it. We'll go into that momentarily, but.
So you mentioned that geometry doesn't matter, but the position. So now we see like conical shape enhanced area. So what does it mean in terms of biology? Would you say?
So you mean this what does this triangle mean? So this basically means that there's a gradient. So probably this triangle is defined by a module of genes that's expressed more highly in these cells. I'll show you how to do this in a second, but we can just look up what those are. So what it seems is that there's some sort of gene module that has increasingly high expression, that there's actually a gradient of expression of this gene module, and the tip of the triangle is like the most high expression of that gene module. So when you see like spikes or cones like this, usually that means there's a gradient of expression of a gene module, and the tip of the cone is the highest expressing. But in general, there's no hard rules about what these things mean, so I think you'll probably develop an intuition from using the tool. Any other questions? All right, so now let's talk about selecting cells. So the main way you're going to, besides just, or actually before I do that, well, yeah. So the main way you're going to, actually no. So there's, besides genes, there's also, there's also categorical labels. So if you see where it says cell type, and there's a button here, if you press that button, it will color the cells by cell type. And if you click on this menu, you can see a couple other color tracks. Cluster index and cluster label are basically the same as cell type, but the other interesting one is replicate. So here the colors represent whether the cell came from the original data set that was in the paper we sent out or in the new data that's been added recently. On the right side, there's a legend. If it's not showing the legend for you, then maybe try switching to another browser. That's always going to be my answer to everything.
Sometimes actually, something that could happen is if you open, then the legend is screwed up. So another thing is to just close it. Yeah, so sometimes, sorry. So sometimes if you have opened the browser, you have navigated to Spring, and then you have rescaled the window, then this legend is going to get screwed up on the right. So then close it and scale and then open.
Okay, so now we're going to talk about selection. So selection is the main way that you can interact with the data. So we'll just start by selecting a group of cells. So if you have a Mac, pretty much in any computer, you can hold down Shift and your cursor should change to a plus sign, and then click and you can drag a box around a group of cells and they should light up in yellow. If for some reason that doesn't work, you can also just click this button here, positive select, and that's equivalent to like holding down Shift. And then to get out of that, just go back to drag, pan, zoom. Mm-hmm.
Do you only have a choice of a square or rectangle? Can you draw a loop?
You can't like draw a lasso, but you can kind of like approximate it.
Somehow I have still something selected which I can get rid of. I think it's a cell type that I clicked, and I don't know how to deselect it to get to this.
So so first of all, you can just zoom out. So if you hold down the command, so let's say we have something selected. If you now hold down the command key, you should also see a plus show up. And if you're on a Windows, I would just click deselect over here on the right side. And now you should be able to drag a box and deselect anything.
As much as you can, let's try to ask questions using a microphone.
What's the difference between index and label?
What's the difference between index and label?
They're basically the same. It's index is internally how we keep track of it. So just don't pay attention to index. So also just regarding selections, so is anyone not able to select cells with the box or deselect cells with the box? Raise your hand if you're not able to.
Raise your hand and keep it up for a couple seconds.
So you can also see there's a button in the top left called Deselect All. So if you click that and nothing's selected, everything will get selected. And if anything is selected when you click that, then everything will become deselected. There's an undo, not for selections. Okay. Then and I'll show you why you want to, actually I'll show you now. So why bother with selections? So let's see this spike coming out here. So right now we have these nice labels of all the cell types, but obviously when we first got the data, we didn't have any labels. So we would get plots like this and have no idea what everything was. So let's say we want to know what this orange spike out here is. Then we would select those cells and then click Show Enriched Genes in the top left. And what will pop up is a list of genes that are on in these cells. So it seems like keratins are being expressed in these cells. Like at any point, if you can't replicate any of the things I'm doing, just raise your hand and hold it up. So when you do this thing, when you select some cells and then click Show Enriched Genes, what shows up in the list is the genes that are enriched in the cells you selected compared to the rest of the data set. You may be in a situation though where you see that, for example, there's a certain tissue and the tissue seems to be kind of in two halves or something, and you want to know the differential expression between the two halves. One second, but you don't. And so in that case, you can actually specifically do differential expression. So you select one group of cells, and then you negative select another group of cells, which I'll explain in a second, and then ask for enriched genes. It'll show you what's enriched in the yellow versus the blue. And for negative selection, you hold down Shift and Escape at the same time while drawing a box. Or if that doesn't work, or if you have a Windows, then just click the negative select button on the top right. In general, these buttons on the top right is one way to switch between the different selection modes, and then the keyboard shortcuts is another way to do it. Are there any questions about any of it? Mm-hmm.
So when the data that you present there, so for example, cordon is showing us 8 there. Does that mean that it's 8 times higher in the selected versus the non-selected, or what is the different numbers there mean?
So when you just select cells in yellow and you ask for what's enriched, the numbers next to the genes is telling you the average z-score, the average number of standard deviations away from the rest of the data is the group of cells you looked at. So if on average the group of cells that you selected are expressing a gene 3 standard deviations above the distribution for the data set as a whole, then you'll see a number 3 there. And when you do differential expression, it's showing you the difference of standard deviations. So if the yellow selection is 2 standard deviations above and the blue selection is 2 standard deviations below, then you'll see a 4.
And so maybe I can follow that question by asking, I guess I should probably maybe know the answer to that question, to the question I'm asking. It's just showing my ignorance. But at which point, which numbers would we actually, presumably if you scroll down, it'll continue to go down. The numbers will go down further and further and further. At which point is it no longer really a difference that we can actually believe?
In general, I wouldn't recommend using the numbers to decide. I would recommend using your intuition and just looking at it. This Spring doesn't really offer a formal test of differential expression. It's mostly like a kind of intuitive visualization tool. So if you wanted to, near the top, everything is going to be obviously on in one and obviously off in the other. Once you're at the point where it's visually ambiguous and you want to know if there's a statistical enrichment, then you should just download the expression data yourself and do a statistical test. And I'll show you how to download expression data in a little bit. Is there a question?
I have a question about negatively selected genes. So just when you enrich, you compare to the total population, but if you want to compare two populations, how do you do that? How do you get the numbers if you want to compare 2 populations? Let's say green to blue, yellow and blue. Can you do that?
So if you select some cells in yellow and some cells in blue, it is only showing the comparison of those 2.
I see. So the numbers would show the comparison of the 2, not not relative to.
No. Yes, it shows it's the difference of their respective comparisons to the whole data. That's true. So in general, just don't. These numbers here are not very important. I mean, this list is really just like a way to put stuff on the screen, and then you just look at what's happening to form an intuition. In general, I wouldn't pay attention to these numbers.
But there would be a lot of difference between if you use one population of cell and compare it to different areas on your plot. So you're saying when you choose 2 areas, then the numbers reflect the difference between the 2 areas, right?
When you pick 2 areas, our intention is to show genes that are specifically enriched in one and like depleted in the other. And behind the scenes, what's happening is that what we're really comparing is their selective. We're asking about the difference in their enrichments to the rest of the data, which may not be the best way to specifically ask. But on an intuitive level, if you do blue select and yellow select, you're asking for differentially expressed genes between the 2 populations. And if you just select yellow, you're asking for enrichment compared to everything else. And what's happening behind the scenes is a little more kind of a hacky version of that.
So I just want to make sure that everyone understands the difference in 2 ways of selecting differentially expressed genes. Just the yellow against everything else, and the yellow against the blue. But no questions about that.
Okay, so another thing you can do is select by cluster. So for example, let's say I freshly arrived here and I want to know where endoderm is. I guess you could look up a marker gene for endoderm, but another thing is you can just click, you can go to the cell type menu and click endoderm, and it will select endoderm cells. And also I added this animation, this popping animation, so you could kind of find where it is on the plot. You can also negatively select a cluster, just if you hold down Escape and Shift, or you click this button for negative select, and then you click a cluster, it'll negative select that cluster. So clicking these clusters over here is another way to like grab onto a group of cells.
Notice the number of cells is marked to tell you how many cells there in the selection.
What?
I'm just pointing to the number of cells.
Oh, up here?
Well, no.
Oh, in here. Mm-hmm. Yeah, and if you hover over this, it'll tell you the percentage. So for example, let's say I select this. Let's say I select this whole bit over here, and I want to know if, and then you go here, it tells you what percentage of each cluster is being contained in that selection, just by hovering over these numbers.
It seems like a low number of endoderm cells.
Yes. Why are there so many 1s and 2s and stuff?
No, I think it says overall there are a few endoderm cells. Is that your point?
Yes.
How many cells are at stage 8? I mean 12, presumably. That's what you're wondering. Out of the X number of stage 12 cells that now were sequenced, how many were endoderm, right?
And maybe it just seems like there should be more.
Yeah. So for example, here endoderm is like less than 1% or something. And I agree. That was one of our initial surprises from the data. Here, can we get a microphone?
No, absolutely.
So, I mean, one of the goals of this will be to try to identify different clusters or different cell states. So maybe you're going to get to this, but because you only have the option for a square, and it's not sort of, so how can we go and try to identify groups of cells that we think might represent a unique state?
I think you just kind of eventually use the square creatively. Like, let's say I want to get a circle right in the middle of here. How would you do that? Well, first I might come at it from the left. Then I might come at it from the left again. Then come at it from the bottom right. And eventually I can get kind of a circle in here.
Okay, got it. I didn't see that part. Good.
Finally, we're going to go a little more into this later, but once you've selected a group of cells, you can actually move them around. So actually, let's do that later. So does anyone have any questions so far? Yeah?
So these numbers, they just represent the numbers of cells you picked up in total. It's not like somehow related per embryo, right?
It's the number of cells, it's just internal to the spring plot. It's the number of cells from that category in the spring plot.
Just maybe to add to Aaron's question, you can also shrink the size of the dots if you want to be more precise in selection.
Yeah, thanks for pointing that out. So there's another, you may find this useful. Up here, if you go to the settings menu, you can change what the visualization looks like. So you could change the size of the dots. You could change the opacity of the dots. The one I use most often is to change the background color. And yeah. Any questions before moving on?
Could you say one more time? So if you were to pick two different lineages and want to compare them, how do you do? How would you? I'm sorry. How would you compare two lineages? So you select two lineages, and that's the number that comes up when you say show enriched genes. Is that the comparison? So if just pick two lineages, is that what shows up?
So if I take these two lineages and I select one of them, then what comes up is just the genes enriched in that one lineage, whether or not they're also enriched in the other lineage. But when I negative select the other lineage, now it's going to come up as genes that are specifically enriched in this one and depleted in that one, in the other one.
It's that way around. How can't be some that are enriched in that one and depleted in that one?
Can you switch the order? Is that the question?
No, so you said that those are ones that are enriched in the yellow ones and depleted in the blue ones.
Yeah.
What happens about ones that are enriched in the blue ones and depleted in the yellow ones? Do they come up on that list too?
No, you just have to switch. You'd have to reselect them the other way around.
Okay.
So to facilitate the collective annotation of this data in the group, we built a tool called Sticky Notes, and these are going to be really instrumental. I really hope you make use of this tool because it's going to be our record of everyone's thoughts about the data. So if you go to Tools and then click Sticky Notes, you should see, it looks like someone already made one. You should see a window that looks like this. This person has misused the tool, whoever it is. So now this is good we're having this intervention. So the author of the sticky note did a great job putting text in it, which is one of the key goals of the sticky note. But there's another aspect to the sticky note, which is that it's supposed to be associated to a group of cells. So let's see. I'll make my own. So here's some cells. I don't know what they are. Let's say that they're CCNO, they're FOXJ1, everyone's favorite gene. So I'm going to make a new sticky. I'll say these are my fave FOXJ1 cells. Okay, now the point is I need to mark that these cells are associated with this note. So with the note, right now the note's deselected. Now I'm selecting the note, and then I'm clicking bind cells. And now the cells are associated with this note. So what happens, for example, is now if I click on it, the cells pop, and that's how you know which cells they were talking about. If I refresh the spring plot, this note is going to disappear. So with the note selected, I need to put in my email address and click Save. And now if I refresh this plot, hopefully the note will still be there.
And everyone else looking at this data should also see the note. Okay?
No, just kidding. It disappeared.
How did you get a new box to write something in? I only see this box that was there.
You go to Tools, Sticky Notes, and then click New.
Oh, new. New.
Oh.
Do we see every sticky note that everybody puts in there?
Yeah.
That's the spirit.
It's going to be like 1 million sticky notes by the end of this. How do you find your own?
You just have to scroll through it until you find it.
Wait, so theoretically, we're not going to be all working on the same set of cells. This set of cells is just something to start you off with. You will soon see how to select some subset, and then you will fork it off as some subset you are working on with maybe 1 or 2 other people, and you're going to be sticking notes on that one.
I actually just realized so I literally just finished making this yesterday. And I just realized that if everyone saves at the same time, then everyone just overwrites each other's attempts to save. So actually, don't make any sticky notes on these main subplots. Only do it on your own string plots, and I'll send a message in the Slack when I fix this problem. Okay, but in general, modulo that caveat, which is going to be fixed in a couple hours, this is the essential principle of the sticky notes.
How do you make your own subplot for yourself so you can work on it?
Yeah, so that is now what I'm going to talk about.
Can you go over again? To make your own subplot.
Can you just repeat again how we copy those things in the sticky notes? Do you copy your genes or you? I didn't follow.
So a sticky note. So here's the procedure to make it. So the situation where you want to make a sticky note is where you found some cells, you think they're interesting. We called them pronephrose, but actually it's some other tissue. Or you see a gene expressed there that you don't expect to be expressed. So now how do we record this observation? So step 1 is you need to have the cells selected in yellow. Step 2 is you need to click New and get a new sticky note. Step 3 is, and just to make sure, with your favorite cells selected, click Bind Cells. So now you have a new sticky note. You've associated the cells to the sticky note. Now you write stuff in it. Then you have to put your email, and then click Save. And in theory, it's now saved. Mm-hmm.
Can you select cells based on gene expression? So you have your favorite gene, you show where it is, and you've already showed us how to do that. But now what I want to do is select all those cells that express that gene. How do you do that?
Yeah, so for that, you use a tool called the expression selector. So let's say I want to select all the cells expressing SOX2. First, I have to show SOX2. Then I go over here to this gray box above the slider and click that. And now a yellow rectangle will appear. That rectangle defines the upper and lower bounds on the range of possible SOX2 expression within which cells will be selected. So if I want to go up to the highest level, I do that. And now the SOX2 cells are selected. And then you just click this to get rid of it, and you can deselect the cells if you want.
I would like to check, is there a way how I can check whether two genes are co-expressed and color them, like SOX2 in green and then the next one in red, and check the overlap and heterogeneity of the expression? I tried the right bits, but this doesn't work with the gene sets and custom colors.
So in this browser, there's the best way to do that. So let's say you see, you want to know among the cells that express SOX2, do they also express SOX3? So I think the best way to do that is first select all the cells that express SOX2. Now you can actually drag them elsewhere. And you just click and drag on any selected cell, and it'll move all the other selected cells. And once that's done, then we just could look up SOX3, for example. And yeah, so these cells are co-expressing SOX2 and SOX3.
This is not safe, this pulling away, not that I change your spring blot.
So you can save, there is a way, if you change the coordinates, you can, we'll go over that in a second, you can save them, but you can't, because this is one of the central main data sets, you can't save it because we've basically turned off the permission to do that. I think that you can't. Let's try. Yeah. Any other questions? Yeah, it looks like they can't. Mm-hmm.
Can you show us how to drag that set away again?
Yeah, so if you want to, say you have a subset of cells and you want to drag them away, so one way to do it is you just have to find any selected node and hold, put your mouse over it, click and hold down, and while it's being held down, just same way you're you would drag an icon across your desktop, you just drag a cell. There's also a kind of expedited way to do this, which is if I have a bunch of cells selected and I just want to move them to the side, you can go to Layout Tools and then Move Left, and it just moves them off to the left of everything else.
We get back.
You need to deselect. You have to select everything.
So if you go to Layout Tools, Undo,
yeah, there you are.
Then it'll just move it back to the last place you had put it.
But undo all?
You cannot take them out. Oh, yeah.
Does it just step piece by piece?
So if you just keep moving cells over and over and over again, undo will just keep stepping back through all the changes you made. Refresh is also the fastest way. Also, this is just a gimmick. I don't know I doubt any of you will use this, but you can also rotate cells and make them and scale them. But yeah.
Say I select a group of cells. I write a note and say, these are obviously, from some other source, whatever. Now somebody starts playing around with that general area. They won't know that I had a sticky note in that area, will they?
No.
So there's no way of alerting them that you are walking on somebody else's sticky noted area, which might be of interest, you know.
The sticky note is not associated to the area, it's associated to
The cells.
the data set. So you
No, I don't know. Yeah, fine. In the next couple hours, I'll make it so if you have cells selected, those sticky notes will be yellow. Oh, by the way, as an aside, the cells are not you can have one cell be part of two different sticky notes. So if you make a sticky note about a group of cells, you're not like taking those cells away from someone else's sticky note.
But I mean, the question, I guess, is as I start messing around in some area, it would be nice to know if somebody else has.
Yeah. Okay. So by the end of today, it will be the case that if you select cells that are associated already with one of these sticky notes, then when you've selected it, that sticky note will have a yellow outline or something.
it is already the case that people could see all of the sticky notes associated with this data set.
Yeah, but you don't have no way to know which sticky notes are associated with your cell type.
Click through the notes and see.
Sure. So
if you just go to Tools, Sticky Notes, and click it, you will see all of the notes made by other people for this big data set.
Yeah, for the data set, but the question is, my question is, if somebody else starts, if I select that area of the political gland, I'm not going to select it precisely the same way somebody else selected it.
Yeah. I'm going to make it, so as of tonight, it's going to be the case that if any one cell that you've selected is in a sticky note, that sticky note will be yellow.
Okay, Caleb.
So outliers has to do with the bioinformatic process that we went through to generate the clustering. So basically, the clustering algorithm that James used will only make a cluster if it has more than 10 cells. And things that just couldn't be part of any cluster were called outliers. But these outliers potentially is a treasure trove, like germ cells is in the category called outliers.
So we should look at the outliers too. Is that your?
Mm-hmm. In fact, if by the end of this, people could just say, if we could carve out cells from the outliers and say, this is this state, this is that state, that would be a very productive, thing to do during the Jamboree.
Quick question. Can you use expression selector for both positive and negative and then compare them?
No, not currently.
I actually try to do it by selecting the very low or kind of negative cells, and then you can drag them and negative select, push them back. I mean,
yeah, I guess right. So you could use the expression selector to get some cells and then move them to the side and then negative select them once they're on their own.
This is an answer to your question, Sasha, right? You first use positive selection through expression to select low expressing cells. Once you selected those cells, you will reselect them as negative.
I've got a question to the gene annotation. So you've got this, this is probably to Leon. Leon can probably address it. So a lot of the Xenopus genes are labeled as LOC genes. And I've seen now that you've got then also additional names. But have you basically re-annotated those LOC names with?
Yes, yeah.
Okay, so this is a.
So most of LOC names have another thing on the right of the gene name, which is the quote-unquote Peshkin name. But let's for now focus on visualization, and we'll talk about gene symbols later.
It's an important point, because if you defined a group that you want to look at more carefully, then it's good to know that these LOCs have actually been annotated, I guess, on the basis of the human genome.
Yeah, about 10,000 of these LOCs have been re-annotated by homology to human genes. And so when you search for a familiar gene, it might be LOC in the genome, but you will find it because we have re-annotated it here. So just start typing it into the gene search box and it should pop up.
Okay, so now I'm going to talk about making a subplot.
Actually, before you do that, so as we teach you to create subplot, I guess it's okay to create a couple, but I'm worried that people will start creating subplots, everyone, and we will sort of make a pile of useless subplots altogether. So just be careful. Every time you create a subplot, you create an entry that everybody else could see on the main page. Okay.
So Leon, is there a way that we could, you're playing with a subplot.
You can save it. I'll show you in a second.
If you make a subplot,
It's automatically saved.
button that says save or not save, so that we can play with making subplots?
Yeah, I'll address this in a second for everyone.
So once you created a subplot and it's a separate new tab, it means it has been saved and there's an entry. But you can delete it later. Just make sure to identify it well when you give it the name and your address and everything.
Well, you're going to talk about that, but it would be better, I think, to have it not saved and only saved if you can save than to have to delete it. Because then I think you like to delete it.
Yeah.
I'm really used to saving things. You type something and save, type something and then you unsave.
Yeah, maybe we'll have an informal system. Maybe we'll make a Slack channel called "I want my subplot to be saved," and if it's not there by midnight every night, we'll delete it. For now, we'll have that be the solution. So anyway, so first of all, why make a new subplot? So there's really two reasons. So when I say make a new subplot, I mean take a group of cells and make a spin-off spring plot entirely just consisting of those cells. Now there's a couple reasons why you'd want to make a spring subplot. The first reason is because now it's your own little playground. You can leave sticky notes galore, change the coordinates, save the coordinates, so on and so forth. And so that's one. Two is that the whole pipe, so when you make a new subplot, what's going to happen is that it's going to take the group of cells you selected and go down back to the raw data, the UMI counts associated with those cells, and entirely rebuild the spring plot from scratch. And there's just, for some reason, these algorithms have a kind of a limited bandwidth of complexity that you can see at any one time. So what happens is that if you make a subplot from a subset of cells, the kind of budget of complexity is now all being targeted to those cells, and you can often see more features or more structure than you could when the cells were co-embedded with the rest of the data set. So that's another reason. The last reason to make a subplot is because that's the best way to combine multiple time points. So you can make a spring plot starting from here. So I'll show you both, and it's pretty much the same either way. So to make a subplot, you select a group of cells, then you go to Tools, Make New Subplot. You need to put some sort of name, which doesn't have any spaces in it and stuff like that. So
does it break it if you put space in?
It'll just tell you not to. Then you put your emails. And then, okay. Then you click Submit. These are some parameters that hopefully, I just don't think it's worth going into. Just don't change them. The one that you could change is Save a Layout Animation, which is just a pure gimmick, but I enjoy it, so we'll do that. So then you click Submit, and it's going to start to make a new subplot. We can come back to that in a second. You can also start from the tree browser and select any group of clusters. It could be across time points, and then it's the same thing. It's exactly the same thing.
How do you visualize those subplots?
So when the subplot is done, there'll be a link here, and you just click on that link, and it takes you to the subplot. Now, in general, okay, so now this is where we were going with saving and deleting. So from anywhere in this whole Spring universe, you can go to Tools and go to All Subplots menu, and that's going to take you back to this home page. And if you scroll down, these are all the subplots that people made. So in the next 10 minutes, I'm going to have everyone make their own subplot, and suddenly we're going to add 20 boxes here, and that's going to become cluttered. So we're going to make this informal system of saving.
See, there's a delete button on each box. Please try not to delete other people's subplots.
So just to complete the demonstration, like here, it finished, and there's a link here. So if you click on that link, then it takes you to the new subplot. Mark had a question. There's a question back here. Oh. Yeah? Oh, okay. Any questions about making new subplot?
Does that have the time element in it, the developmental staging?
So if you made one, so because these cells are all from one stage, then yeah, they don't. But if you make one from the tree browser, then it will have time point as one of these options.
But
can you combine different subgroupings?
If you've made two subplots, then there's no way to kind of explicitly take the union. I would just go back to where you started and do it by hand.
What about combining two different time points? But I don't want to use the tree view. I want to go to stage 12 and stage 13, select two lots of cells, and then kind of explore them together. Is that possible?
There's currently no way to do that. Oh, sorry. One thing you could do is you could you could just use the tree browser to make like a bigger than necessary group, like a strict superset of what you want, and then go into that subplot and make a subplot.
If you already have the home page, quote unquote, open, and you created a subplot in a different tab or different window, the home page is not going to automatically refresh to show you your newly created set. You need to refresh the home page for it to show the new set, okay?
I'm going to select from the old stages.
You can, yep. So it's just kind of slow, but you can go in, anything I just showed you applies over here, which is just a massive spring plot with all the stages together. It's not even loading on my computer. So, but anyway, yeah.
So for the temporal, related to Aaron's question, so can you select in the tree plot, select different stages and then deselect the stages you don't want and then create a subplot? So then you only get stage 12, 14, whatever.
Yeah, I mean, you don't have to deselect. So if you just want stage 12, I just selected all of stage 12.
But if you want to jump around stage 12 and then stage 18, I don't want anything in between.
Yeah, then you just go select stage 18 too.
Oh, okay, just one particular branch also.
I mean, yeah, in general, you just select the things you want and don't select the things you don't want, and it'll just only the yellow stuff will go in.
So somewhere there is an optimal level of complexity, I want to say, between the giant octopus-looking plot where we have all of the stages all in one spring plot, and the degenerate subplot with a single cell, right? There is some level of granularity where we want to stop for our encyclopedia of cell types. So this is what we're going to be doing. Maybe iteratively split one stage into 2 or 3 descendant of germ layers and then split it some more and annotate.
I guess, yeah, I kind of ended up, all right, whatever. So there's one more thing I want to show you guys before we pause for the you do it yourself part, which is something called dynamic mode. So this gives you a more intuitive handle. I think I just crashed my computer by trying to load all stages. Okay, so if you go to Tools, Dynamic Mode, this is going to take you to a kind of older version of Spring. So please don't stay here. But it has the advantage that if you click Resume Dynamics, now it's actually showing you a live simulation of the force layout. And this can be useful for trying to intuitively understand the structure of the graph, like beyond just the 2D coordinates that got generated automatically. So you can, for example, select cells and move them around. And you'll often see that the structure appears to kind of rotate. You can get a sense that the structure you're looking at really does exist in a kind of higher dimensional space, and we're really forcing it into 2 dimensions. Sorry?
Get that to move.
So you go from normal, you go to Tools, Dynamic Mode, and it will open a new tab. And then you click Resume. Up here, it used to say, it will say Resume Dynamics. You click that, and then everything should start moving. Here.
But then, can you use this to have a better view, like froze it in a more spread out view than the original ones?
Yeah, so if you say you create a layout that you like here and you want to keep it, then go to Layout Tools, Save Coordinates. And then when you go back here and refresh, it'll show the new coordinates. And actually, in general, when you have your own subplot, if you do anything you want to do, move this out here, move this out here, in either this mode or the dynamic mode, if you just go to Layout Tools, Save Coordinates, then it will save it.
So why don't we start with everyone doing exactly the same one simple thing and make sure that everybody can do that, and then let people do their own exercise. So maybe like start with stage 14, which has a bunch of cells and a little bit of complexity, not overwhelming. Select subpopulation of cells based on SOX3. Create your own new subset. Call it with your name, and so raise your hand if you have trouble. We will make sure that everybody can do that.
And make a sticky note about it.
And make a sticky note about that.
Like within, once you've made your own subplot, go navigate into that subplot and make a sticky note about some cells.
How do I get out of this dynamic mode again?
Go to Tools. So first of all, it's in a new tab, so the old tab should still be there. But also, you can go to Tools. Or once you're in dynamic mode, if you go to click Go To, and then Static Mode, it'll send you back to static mode.
So I put an exercise up here. You go find stage 14. Select cells expressing SOX3, create a subplot, and add a node to it.
Question. Expression selector. Why does anything happen if I go here? Oops, sorry, I just wanted to move the yellow thing. If I go here at all, isn't that so?
You're selecting cells expressing in this range.
Right.
So you're increasing the range.
So, okay, so there's still cells. So green just tells me.
If there is this number, it means there is a cell expressing this high.
Okay.
Okay, this shouldn't take more than 2 minutes, all you guys.
Well, I screwed up because I made it a new plot. So
You've. No. Did you click Process yet? Oh, go down. If you move it up, or scroll down, move the whole window up. So if you click it and drag it up.
So now,
do you know what you called it?
Here, I'll show you. Click this one.
Did you go delete it already?
Just this itself. Oh yeah, you did it. So there's the link.
Then you scroll down and find your. Yeah. What do you call it? Okay. Right there. So delete it.
Then I'll see the link here.
Page 14.
Yeah, somebody else actually has done the same thing I did. So now, yeah, let's go back to here. And now I've
Close
selected.
this one.
It's running, so it's fine.
So you
How do you get from the dynamic mode to
Done that. I've already
selected.
done that.
Okay.
So now I just need to do, so I
same
need to
thing.
do the sticky.
Make new.
So right now you're in regular mode.
Now it's doing something. No.
So right now you are in regular mode.
Yeah.
If you
Oh, okay. I'm sorry.
say here you go to dynamic mode.
You deleted,
Oh,
do the right thing.
so this is regular.
But you need to add a sticky note to that.
Oh, now I need to add it.
So the last step of this exercise
This is just another tab. This
sticky
is all
note
of stage 10. But
within your subset.
point is that once you go,
Not
if
to the
anytime you
whole
go to
stage
dynamic mode, it'll open a new
set,
tab.
but
So you just
within
close that
your
tab.
subset, okay? After you created the subplot, you annotate that subplot.
So, okay, so I had done the right thing.
What's the trick for selecting based on I highlighted genes.
Highlighted by what?
So if you type in SOX3, you get these green cells.
Yeah.
There's a way to select just the green cells.
Yeah.
I just dragged it.
Can you
Well, I don't know how.
let's.
It's doing this.
Yeah, that's what I did.
Once you select it,
You need to give it a name here. And I put my initials, because otherwise everybody's going to say the same thing. So,
That was just the title of this thing.
but then it's loading.
Okay, so now.
Gonna drag that. Yeah.
We have to wait to run before to do the sticky notes?
Yes. Once you selected cells and created your subplot, it takes a few minutes for it to actually get created before you can annotate it. Can you take that?
They told me run complete, right? And then I was checking the email.
I know if it's saving or not. I created
And you click save.
Yeah, I tried, but it's saving or?
I think
That's
so.
Never
Let's
worked
try.
for me.
Is this one of these yours?
Yeah, both of them are mine.
So there's no direct feedback if it's working.
Oh, okay.
It should just work every time.
I see. So this is fine.
Yeah. So actually, the one situation in which it won't work is if you don't put an email address, but you can see because it'll go like that.
It shows all of the sticky notes.
Oh, where all the others?
Oh, 1 second. Sorry.
Once you have made a new subplot,
Yeah.
you get rid of.
This set, which hopefully you just created.
Oh, drag it back down.
I have one other question.
Oh, yeah.
Those cells you edit your note
Yeah.
So if I go back to the previous note, so I go back to this note, I'll find the note, I have to go to Slack to find the note?
No, you just go to tools. The bottom, sticky notes.
Sticky notes, yeah.
Is that your note?
Yeah.
So if you just type some text and then click save, it'll save the revised version.
How do I know when it's
Yeah.
But a previous sticky note I did.
How many have you done? And so you must not have saved the first one. It should be that a comprehensive. So let's try making another one.
And how do you know there's?
It's okay. I want to select this group of cells.
Just hold down Shift.
Also, might take longer.
Maybe it's just they were using.
I'm sorry?
Yeah.
If I click on it,
And then go to Tools.
I got the subplot, but I can't figure out how to get it.
Sticky Notes at the bottom. Oh, what genes? Yeah.
Here?
Oh, here.
So then,
Let's see. Layout.
Yeah.
You know, I mean, it's amazing. I mean, you just
That's the key feedback loop.
Okay.
So I got one thing.
So how do I know that it's associated?
What's the name should be?
Yeah, you should use space. Just write something like.
This is wrong.
There's this kind of salt and pepper. It's like mouse will go down, but the precise location.
So how did you get this? What you want? Yeah.
You click on the node. So which cell?
You can use that.
So you can go to download.
But I clicked on
you can do it.
That's my understanding. I just go
my email.
I had it on.
You know, just, you just,
I'm using the Gmail. How do I know? The one you are using is like.
So it's not here, but I sent it to your email, right?
You saw your thing for this?
Like a new
I get an email. Yeah.
Sorry.
Oh, yes.
I tried this with previous, I select using this. I can't.
Yeah, so
That's what
probably
it was.
the easiest would be.
Yeah, I know this one. This one works.
Okay. So I would just zoom very far in until you clearly see it, and then.
It was James. Oh, there he is.
Try bright one.
So.
Yeah. I got it. Thanks.
Well, so if you want to raise your hand.
And all these things? Or is that
You
all?
have to.
Send the email when it's.
That's why, because it's in my name.
When you create a node, you don't have to have your email, but when you create a subset, please add your email.
I mean, you don't have to.
Why?
Somebody needs to reach you afterwards.
I'm here now. How do I submit this one?
You just, if you can't see the bottom, just move the window up. And then.
Oh, then there's a submit button.
Yeah.
Yeah.
So it's a little too, window's a little too big for everyone's screen.
To highlight that these are secure cells. You see that one's missing actually. So, and then I
Several minutes.
saved it and now.
There's one question we get over and over again. I created a note. How do I know that it actually got saved? So you close this note, and then you go back to the menu to Tools, Sticky Notes, and that shows you all of the notes created for that set. And then once you click on the note, it will flash the cells which this was bound to.
But the true test of whether it's been saved is if you refresh the page and it's still there. And if any of you, if anyone finds that it's not saving, that is, could be just because the software is messed up. So please let me know.
If you are done, go get coffee. This way we will know how many people are finished.
These ones are big, and the other ones
So
are
you
small.
can go layout. Settings.
I know, but only the.
Oh, that's cool. Sorry, I broke. Yeah, this was cool. Can you recreate this? Hey, Caleb, there is a cool bug. He was able to make some of the cells bigger than others.
Deselect all.
This,
Okay, now select new one.
take a look.
Now you have a new set, new note.
Oh, associated with the sticky note. Nice job.
So do screen capture, I guess.
Yeah.
If I go here.
Yeah, see? Interesting. If you could click there.
Yeah, so it doesn't go back there, then these ones appear. No, it's fine.
I'm not sure if this is my browser. To save every time.
Oh, we closed but didn't save.
I then reload the plot.
Yeah, I would just, so I think really what's happening is that. I thought you grabbed a chair. So I'm sure when you reload this.
I can go to the restroom after this.
Yeah, so I should add a slider that looks.
Page 14.
All right, just.
Select the genes that are with SOX 3. Huh? So these are these here, huh?
No, you haven't selected anything yet.
I basically highlighted them, yeah?
You did, but you haven't selected the cells.
Okay, so I have to select them now. With this tool here? Or shall I go here to the expression selector?
Enough people are having trouble that I think it makes sense for Caleb to do this exercise on the screen for everyone.
With notes?
Just demonstrate step by step. Go to stage 14, select cells expressing SOX3, make them into a separate subplot, and attach a note to it.
Okay.
If you're not done by now, you failed.
I just made it under the wire.
Okay, so for anyone not under the wire, here's, all right, so I've gone to stage 13. 14. Oh, I apologize. Yeah. So here I am in stage 14. And where it says enter a gene name, I just click that and I'm going to type SOX3. Okay, everything should turn black and green in a second. Interesting. Okay. Okay, now I click this box, expression selector. Now I have a group of cells. Then you go to tools, make new subplot. So this is something that people have been having. Sometimes it's down here and they can't see where the submit button is. Then you would just move it up. And sometimes it's up here and you can't see where the close button is. So just move it down. Okay, then you would click submit. I'm not going to do that because then it'll take a couple minutes for it. I mean, yeah, so then you click submit and stuff down here will start appearing. Eventually a link will appear here that says click here to get the new subplot. Let's pretend that already happened and we'll just go to one of these. So once you click it, you should end up somewhere like this. Then you go to tool. Is this too fast? Am I doing okay? Okay. Then you go to tools, sticky notes. Type something. Second, select some cells and bind the cells. And then last, put your email.